Welcome to this lesson on nucleic acids. Today, we'll explore DNA and RNA, the remarkable molecules that store and transmit genetic information in all living organisms. These molecules are fundamental to life as we know it, carrying the instructions needed to build and maintain every cell in your body. Nucleic acids are biological macromolecules, which means they are very large molecules essential for life. Their primary function is to store and transmit genetic information from one generation to the next. Structurally, nucleic acids are polymers, meaning they are made up of many smaller units called nucleotides that are linked together in long chains, similar to beads on a string. Every nucleotide, the building block of nucleic acids, consists of three distinct components. First, there's a 5-carbon sugar called a pentose sugar, which can be either ribose or deoxyribose. Second, there's a negatively charged phosphate group that helps link nucleotides together. Third, there's a nitrogenous base, which comes in two main types, purines and pyrimidines. These three components work together to form the basic unit of all nucleic acids. The nitrogenous bases fall into two categories based on their structure. Purines have a double-ring structure and include adenine, abbreviated as A, and guanine, abbreviated as G. Pyrimidines have a single-ring structure and include three bases: cytosine, or C, which appears in both DNA and RNA, thymine, or T, which is found only in DNA, and uracil, or U, which is found only in RNA. These bases are crucial because they carry the genetic code. DNA, or deoxyribonucleic acid, is often called the blueprint of life. It has a distinctive double helix structure, consisting of two strands that twist around each other in opposite directions, which we call anti-parallel. The bases pair in a specific way. Adenine always pairs with thymine, and guanine always pairs with cytosine. DNA contains deoxyribose sugar, and its primary function is to store genetic information for long-term use, passing it from cell to cell and from parent to offspring. RNA, or ribonucleic acid, serves as both a messenger and a worker molecule in cells. Unlike DNA, RNA usually exists as a single-stranded molecule, though it can fold back on itself. In RNA, adenine pairs with uracil instead of thymine, and guanine still pairs with cytosine. RNA contains ribose sugar, which has one more oxygen atom than deoxyribose. The primary functions of RNA are to transfer genetic information from DNA and to participate directly in protein synthesis. There are three main types of RNA, each with a specific role. Messenger RNA, or mRNA, carries the genetic code from DNA in the nucleus to the ribosomes in the cytoplasm. Transfer RNA, or tRNA, transports individual amino acids to the ribosomes during protein synthesis. Ribosomal RNA, or rRNA, forms both the structural framework and the catalytic core of ribosomes, which are the protein-making factories of the cell. All three types work together to produce proteins. Let's compare DNA and RNA to understand their key differences. DNA is double-stranded, while RNA is single-stranded. DNA contains deoxyribose sugar, whereas RNA contains ribose sugar. The bases in DNA are adenine, thymine, guanine, and cytosine, while RNA uses adenine, uracil, guanine, and cytosine. Finally, DNA is more chemically stable and can last for long periods, while RNA is less stable and typically exists temporarily in the cell. DNA replication is the process by which cells create identical copies of their DNA before cell division. The process occurs in three main steps. First, an enzyme called helicase unwinds and unzips the double helix, separating the two strands. Second, free nucleotides in the cell pair with the exposed bases on each strand, following the base-pairing rules. Third, an enzyme called DNA polymerase links these nucleotides together, forming two identical DNA molecules. Each new molecule contains one original strand and one newly synthesized strand. The central dogma of molecular biology describes the flow of genetic information in cells. It begins with DNA, which stores the genetic code. Through a process called transcription, DNA is copied into RNA then, through a process called translation, RNA is used as a template to build proteins. These proteins perform most of the work in cells and determine an organism's traits. This flow from DNA to RNA to protein is fundamental to understanding how genetic information is expressed in all living organisms.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.